Usually when we talk about biological wiring, there's this expectation of like clinical precision.
Right, yeah, like it's engineering.
Exactly.
Yeah.
You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is. It's binary, it's visible, and honestly, it's comforting.
Oh, totally. We like to think of our biology as having a very rigid architectural blueprint.
But then you step into the world of our senses, specifically the sense of smell, and suddenly that x-ray machine feels like it's completely broken.
Yeah, it really does.
We tend to picture our nose as this chaotic cavern where scent molecules just waft in and bump into random receptors. But there's a groundbreaking new paper from Cell by Brann and colleagues that just proved that image completely wrong.
Completely wrong, yeah.
There is a highly precise hidden geographic map inside your nose, and today we are going to completely decode it. We're looking at their 2026 paper, A Spatial Code Governs Olfactory Receptor Choice and Aligns Sensory Maps in the Nose and Brain. Okay, let's unpack this.
To really appreciate what Brann and his team pulled off, we have to grasp the sheer mathematical scale of the olfactory system.
It's massive.
It is. In a mouse model, the genome encodes 11,172 functional olfactory receptors. And we just call them ORs for short.
Right.
And the strict developmental rule here is that each mature olfactory sensory neuron expresses only one of these ORs, like one cell, one receptor type.
So you have a massive menu of nearly 1,200 items, and each sensory neuron gets to order exactly one dish.
That's a great way to put it. Now, under the old scientific consensus, researchers thought the nose was divided into 4 to 9 broad, chunky anatomical zones.
Like buckets, right?
Yeah, exactly. The prevailing idea was that a cell would find itself developing in, say, zone 3, and then it would just randomly draw an OR assigned to that specific bucket.
So it was viewed as highly stochastic, just random within those broad arbitrary boundaries.
Right. But this 2026 paper completely detonates that textbook model. The researchers utilized highly advanced spatial transcriptomics, specifically techniques called stereosec and MERFISH.
And MERFISH stands for multiplexed error-robust fluorescence in situ hybridization. Right.
Precisely. And the brilliance of MERFISH is that instead of grinding up tissue to sequence the RNA, which, destroys all the spatial data, it uses combinatorial fluorescent barcodes.
Oh, wow.
Yeah, so it can identify thousands of different mRNA transcripts while they are still sitting in their exact physical locations in the tissue slice.
It's like leaving the books on the library shelves and reading them where they sit, rather than, dumping them all in a pile on the floor to catalog them.
Exactly. And what they saw when they looked at the shelves wasn't a series of random chunky buckets.
No zones at all.
There are no zones. Instead, there is a remarkably smooth, continuous gradient. Every single one of those roughly 1,100 ORs has a unique, stereotyped mean position along the dorsoventral axis.
So basically, from the top of the nasal cavity down to the bottom. Let me put an image to this for you listening. Imagine a giant piano, but instead of 88 keys, it has 1,100 keys. The old model basically thought these keys were smashed together and tossed into 4 or 5 big buckets.
Right.
But the new reality, according to this spatial mapping, is that the nose is a perfectly ordered keyboard spanning from top to bottom, with each of the 1,100 keys having its own exact sequential address.
That is a perfect visualization of the gradient.
But wait, I have to push back on this architecture.
Okay, go for it.
If you actually look at a cross-section of the inside of the nasal cavity, it is not a smooth piano keyboard.
No, it's definitely not.
It is a chaotic, folded labyrinth of bone and cartilage called turbinates. It looks like a complex cave system. So how can there possibly be a smooth, continuous, 1,100-point linear map on such a folded, irregular surface?
Well, that contradiction is exactly why it took until 2026 to figure this out. The physical tissue is incredibly convoluted, but what the researchers did was mathematically extract the manifold.
Extract the manifold, meaning they used an algorithm to smooth it out.
Yes, imagine taking a crumpled up piece of paper with a delicate color gradient printed on it and computationally flattening it out. When they took the complex 3D shape of the turbinates, an algorithm continuous sheet of geographic coordinates just revealed itself.
So the map was always there. It was just folded up to fit inside the skull.
Exactly.
That's staggering.
Yeah.
So just to give you a quick bullet summary before we move on, we got about 1,100 ORs. The old model said they were grouped in random chunks. The new model proves they are laid out on a continuous, precise, geographic map.
Right, a perfectly ordered gradient.
So next time you smell a cup of coffee, think
Yeah.
about the exact microscopic coordinate in your nose that got that molecule. Does knowing your senses operate on a strict geographic grid change how you think about smelling?
It really should. And realizing that grid exists immediately forces the next biological question, what is driving this map?
Right, because how does a single cell on that crumpled paper know its exact coordinate?
Yeah.
If I'm a developing neuron, I don't have eyes.
Yeah.
I don't know my latitude.
Exactly. The paper reveals that this map is driven by a coherent transcriptional program of roughly 250 genes.
250 genes working together.
Yeah. The researchers calculated something they call a DV score, or dorsoventral score, based entirely on the expression levels of these 250 genes.
So the cell's physical location is quite literally written in its active genetic code.
Yes.
And we're talking about heavy-hitting regulatory genes here, right? Like transcription factors such as N5 and RAR.
Alongside axon guidance molecules like ROBO2 and NRP2.
Okay, so the cell is basically holding a GPS coordinate.
Exactly. But what's fascinating here is the timing. Precursor cells, the stem cells that will eventually mature into these olfactory neurons, express this DV score before they actually pick their specific receptor.
Wait, really? The spatial identity is established first?
Yes, location precedes function.
Think about the implications of that. It's like a highly advanced GPS system being installed and fully activated in a car before the driver even decides what radio station they are going to listen to.
That's a great analogy.
The cell establishes its exact physical location in the universe. And then that location dictates which receptor it is allowed to become.
It profoundly flips our understanding of cellular development in this system. I mean, we traditionally think a cell chooses its identity and then perhaps migrates or adapts to suit its environment.
Right. Form dictates place.
Exactly. But here, the physical geography dictates the cellular identity.
So to quickly recap this mechanism for you, 250 genes create a DV score, which defines the cell's physical position. And those precursor cells know their location before choosing their receptor.
Spot on.
If your cells know their physical location before they know their biological function, it makes you wonder how much of who they become is simply based on where they sat.
It's a profound thought.
But that leads to an obvious mechanical gap. If the precursor cells are plotting their coordinates on this 1100 key piano before they even decide what to be, there has to be something broadcasting that location. What's acting as the GPS satellite?
Well, the researchers pinpointed a powerful chemical signal, retinoic acid, or RA.
Retinoic acid.
Yeah. Retinoic acid signaling emanates from the mesenchyme.
And that's the layer of connective tissue sitting just underneath the epithelial surface of the nose where these neurons are developing, right?
Precisely.
So the structural tissue underneath is essentially whispering positional instructions to the neurons on top.
Exactly. And the team didn't just model this computationally, they proved it with an incredibly elegant ablation model using a chemical called methimazole.
Oh, the methimazole experiment is wild.
It really is a masterclass in developmental biology. So methimazole is a chemical that temporarily destroys the nasal lining in mice. It essentially forces the entire olfactory system to completely regenerate itself from the underlying stem cells.
You've essentially wiped the hard drive and forced a total reboot.
A total reboot, exactly. And during this critical regeneration window, they manipulated the retinoic acid signal.
How do they do that?
Well, in one cohort of mice, they applied RA inhibitors drugs like Win 18446 or BMS 493 to essentially deafen the cells to the RA signal.
Okay, so blocking the signal.
Right. And in another cohort, they flooded the system with excess retinoic acid, specifically all-trans retinoic acid.
And the physical map completely shifted, right?
It did. When they blocked the retinoic acid, the newly regenerating cells overwhelmingly expressed dorsal receptors.
Which are the ones usually found at the top of the spatial gradient.
Yes. But when they added excess retinoic acid, the cells shifted and became ventral receptors, the ones at the bottom.
Wow. So it proved definitively that retinoic acid acts as a continuous master gradient. It is this invisible physical tide that translates three-dimensional space into specific genetic instructions.
That's exactly right.
I picture the retinoic acid gradient like a Wi-Fi signal in a tall building.
Ooh, I like that.
Let's say the router is in the basement. Down at the bottom, in the ventral region, you have 5 bars. Strong signal.
Right.
Up at the top, in the dorsal region, you barely have 1 bar. The cells don't need to consult a complex topological map. They just read how many bars of retinoic acid signal they are receiving.
And that instantly tells them their exact latitude.
Exactly. So just to summarize this piece for you listening, retinoic acid is the master ingredient, acting like a biological Wi-Fi signal. And manipulating it with drugs actually alters the physical sensory map.
It's incredible control.
Imagine if a simple chemical gradient dictates the wiring of a system with over 1,000 variables. What other complex systems in your body might be governed by a simple, invisible chemical tide?
It makes you look at developmental biology completely differently.
It really does. But I have to challenge the fidelity of this system. We are talking about an analog chemical tide, a smooth, continuous wash of retinoic acid.
Yeah.
How does a muddy analog chemical gradient dictate digital binary precision for 1,100 different receptor types? Doesn't the Wi-Fi signal get too ambiguous to pick just one specific key on the piano?
That is the crucial question, and it brings us to how biology converts analog to digital. The answer lies in the non-coding regions, the promoters surrounding the olfactory receptor genes.
So the non-coding DNA.
Right. Promoters act as the threshold switches for these genes. The researchers found specific binding motifs within these regions that have varying affinities for the transcription factors triggered by the RE signal.
Okay, so the receptors that live in the dorsal, or top part of the nose, have a high number of binding sites for the transcription factor EBF.
Yes.
Meanwhile, the ventral receptors are heavily enriched for NFI and RAR binding motifs.
Right. So think of it this way. A low-affinity promoter might need a massive flood of retinoic acid to activate, meaning it will only turn on in the deep ventral regions where the signal is strongest.
And a high-affinity promoter only needs a tiny drop, so it activates dorsally.
Exactly. The DNA sequence itself acts as a digital filter for the analog chemical signal.
Here's where it gets really interesting. To prove that the DNA sequence itself dictates the physical location, they used F1 hybrids.
The F1 hybrid design is brilliant.
Yeah, they took two genetically distinct wild strains of mice, one called T and one called C57BL6J. These mice have tiny, single-letter differences in their DNA, known as SNPs, specifically in the upstream promoter regions of their receptor genes.
Right, and when you cross these two strains, the resulting offspring has one copy of the OR gene from the Cas parent and one copy of the exact same OR gene from the C57 parent.
Residing inside the very same cell nucleus.
Exactly.
Which means any transacting signals like our retinoic acid Wi-Fi are hitting both copies of the gene equally. The environment is identical.
So the only difference is the cis-regulatory sequence, the tiny variations in the promoter?
Yes. And when they mapped where these alleles were expressed in the baby mouse's nose, they found something astonishing. The allele from the cancer parent mapped to one specific physical location.
And the allele from the C57 parent mapped to a distinctly different location in the exact same nose.
The exact same gene, just with slightly different non-coding promoter sequences, physically moved its zip code.
It's wild.
The non-coding DNA literally dictates the physical geography of the sensory map. So a quick bullet recap for you here. Promoters dictate a cell's location, acting as keys for transcription factors like EBF and NFI. And those F1 hybrid experiments prove that tiny genetic SNPs actually shift physical locations.
It highlights exquisite evolutionary tuning.
It makes you wonder if just a few nucleotide changes in a promoter can physically shift where a receptor is built. Just how unique is the microscopic geography of your own nose compared to the person sitting next to you?
Right. We all have unique genetic variations, so we might literally have entirely unique sensory landscapes.
And that spatial precision becomes even more critical when we look at the next phase of the system. Because, it's not enough to just catch the scent molecule in the nasal epithelium.
No, the nose has to transmit that data to the brain.
Right, the signal has to travel to the olfactory bulb in the brain, which has its own highly precise spherical map of way stations called glomeruli.
And the wiring problem here is immense. I mean, how do the axons, the long, delicate wires extending from the sensory neurons in the nose, find their perfect matching target in the brain's map?
Yeah, you have 1,100 different types of wires growing through tissue, and they all need to plug into the correct socket in the olfactory bulb without crossing streams.
It's a logistical nightmare.
If I were engineering this, I'd need a secondary routing system, like a whole new set of instructions just for cable management.
But biology is far more elegant. Remember that DV score?
The geographic GPS.
Yes. The mathematical expression of those 250 genes that tell the precursor cell where it is and which receptor to pick. That exact same DV score also controls the expression of the axon guidance genes we touched on earlier.
Like ROBO2, NRP2, and CAM1, and PLXNF4.
Exactly.
Wait, so the exact same molecular score that decides your physical attitude in the nose also packs your bags and hands you the flight path to the brain.
It's an incredible economy of biological mechanism. The physical location in the nose directly and mechanistically determines the wiring to the brain. The spatial map in the sensory periphery is perfectly aligned with the target map in the central nervous system. And both are governed by the exact same continuous transcriptional code.
It's not just a GPS that tells you where you are standing. The address in the nose is the shipping label to the brain.
Right.
One chemical gradient, driving one genetic score, solving three massive developmental hurdles simultaneously: where to sit, what to smell, and exactly where to wire the data.
It really blurs the line between form and function.
So, to quickly summarize this connection for you listening, the brain has a target map of glomeruli. The DV score programs the axon guidance genes, which means the map in the nose directly aligns with the map in the brain.
Perfectly aligned.
Does form follow function? Or does your physical location entirely dictate both your form and your function?
Well, investigating that function brings us to a critical evolutionary stress test of this whole system.
Okay, what do you mean?
If receptors are strictly ordered by their spatial location, we have to ask, do ORs that sit next to each other on this 1100 key piano physically respond to the exact same chemical odors?
If I'm thinking about this from an evolutionary standpoint, putting all your smoke or predator receptors in one tiny millimeter of your nasal tissue is a terrible idea.
Why is that?
Well, if you get a scratch or a localized viral infection right there, you'd lose the ability to smell a fire. Did the data show that nature was smart enough to scatter the targets?
To test exactly that, the team used a technique called ActSec, or activation sequencing.
How does it work?
They exposed awake mice to specific targeted odors and then immediately sequenced the nasal tissue to see exactly which cells fired in response. Crucially, they mapped those responding cells back to their DV score.
So they matched the chemical response to the physical geography.
Yes, and they found what they term weak chemotopy.
Chemotopy meaning organizing the tissue by chemical response.
Right. There is a loose pattern, for instance. Acidic smells tend to activate dorsal glass eye receptors at the top of the cavity, but overwhelmingly, for most odors, the cellular response is heavily distributed across the epithelium.
So they aren't vulnerable clusters?
Not at all. Even when activated receptors share similar DV scores, they are physically separated by 100 to 200 intervening receptors. They are functionally scattered.
If we connect this to the bigger picture, this is an evolutionary masterstroke. By deliberately distributing similar tuning properties across the physical tissue, the system becomes incredibly robust.
Exactly.
A localized injury won't wipe out your ability to detect rotting food or a predator. The map is structurally precise, but the function is strategically distributed.
Yeah, we see this in server farm architecture and decentralized grids.
Right. Nature favors scattered resilience over perfectly grouped vulnerability. Let's do a quick final recap on this point. ActSec measures odor response. And it shows that while weak chemotopy exists, spreading out the receptors provides an evolutionary robustness against local injury.
It's a profound engineering principle, hardwired into a system that we previously thought was just random chaos.
Nature favors scattered resilience over perfectly grouped vulnerability. Where else in your life or in technology do we see this kind of scattered backup system? It's fascinating to think about.
It really is.
So let's pull all of this together. We started with an old model that saw the olfactory system as a series of random chunky zones. But by unrolling the three-dimensional geometry of the nose, Brann and colleagues revealed a precise, approximately 1,100-point geographic map.
A map driven by a smooth retinoic acid gradient acting as a biological Wi-Fi signal.
Filtered by incredibly specific non-coding promoter sequences.
Seamlessly wired to the brain's olfactory bulb using the exact same spatial code.
And scattered just enough to ensure the organism's survival.
This raises an important question, though. We've unrolled the spatial map of the nose, but it forces us to consider how developmental gradients reliably organize thousands of discrete sensory channels in three-dimensional space throughout the rest of the nervous system without chaotic overlap.
It really highlights the hidden, elegant order beneath biological complexity. So what does this all mean for us?
Well, there's a lot to consider.
I want to leave you, our listener, with one final provocative thought that the researchers hint at. We know those precursor stem cells sit right underneath the mature sensory neurons, constantly receiving signals.
Right.
And we know from other studies that odor experience can actually influence the number of sensory neurons that respond to a specific smell.
Yes, the olfactory system is uniquely neurogenic. It turns over and adapts throughout your life.
Could frequently smelling a specific scent, like your absolute favorite perfume, the ambient smell of your local coffee shop, or even the scent of a loved one, actually stimulate those underlying stem cells?
Oh, that's interesting.
Could your daily repetitive scent experiences be actively instructing those precursor cells to alter the gradient, literally redrawing the microscopic geography of your nose over the course of your lifetime?
It strongly suggests that the sensory map isn't just a static piece of paper printed at birth. It's a living document, constantly being edited by your environment and your behavior.
Which brings us right back to our X-ray analogy from the beginning.
Yeah.
When you look at an X-ray of a broken arm, you are looking at a rigid snapshot of the past.
Yeah.
But when you look at the sensory map of the nose, you aren't just seeing where you are right now. You might be looking at a dynamic living atlas of everything you've ever experienced. Thank you for joining us on this deep dive.